And I'm very pleased to introduce Dr. Vesna Jevtovic-Todorovic. She is a professor and chair of anesthesiology and professor of pharmacology at the University of Colorado, Denver. Welcome.
Thank you, Jill. Thank you so much. I'm truly honored to be invited, and all the work that you've done has been really impressive. It makes a huge difference. Let me see whether I can share. Can you guys see this okay in a presentation form? All right. So Jill asked me to start by giving a bit of an overview of what we know so far about developmental toxicity of general anesthetics, and then I will finish with some of the epigenetic work that we did over the last couple of years. I have nothing to disclose. I'm not peddling any products. I'm not selling anything. Our funding comes from private foundations and the NIH, and I will acknowledge the sources of funding at the end. So when we think about pediatric anesthesia, and actually Jill alluded to that a few minutes ago, we all know that the advances in pediatric and obstetric surgery have placed incredible demands on anesthesia. The cases are very complex, they're very lengthy, and we repeatedly anesthetize kids without really having a second thought. Jill is exactly correct that the number of premature births is pretty substantial. It's 10 to 12% of overall birth rate is actually premature babies. And I've heard from some of the neonatologists that they took care of premature babies as young as 22 weeks of post-conception. This is a photo that I got from a very good friend of mine who is a pediatric anesthesiologist at Boston Children's Hospital. This baby is or was 450 grams. That's almost the size of his hand. So as you can see, this little one has a lot going on, a lot of IVs, intubated, most likely sedated, and very likely coming to the OR multiple times during his or her stay in the intensive care unit. So very likely to be exposed not only to sedatives, but to general anesthetics repeatedly. And of course, I'm sure all of you will agree with me if I'm to say that a child cannot be considered a small adult, especially when it comes to the anesthesia management. So obviously, you know, we can spend a good amount of time discussing what it really means to be a child anesthetized and how different that is from adults. But today, I would like to focus your attention on a central nervous system because this is really of great focus to my research. What we know in humans is that central nervous system is not completely developed at birth. We know that it weighs approximately 335 grams at birth, and we know that brain doubles in size by 6 months and definitely triples in size by 12 months. So when you think about the length of human life, that is in average in the United States anywhere between 78 to 80 years of age, you can imagine how complex this time period during our development is. In short 12 months, our brain triples in size. So what happens, what actually constitutes that enormous growth that we often refer to as a growth spurt? So that time period that we always talk about and are interested in is referred to as synaptogenesis. And it is different from neurogenesis that happens during the first and second, in particular second trimester of a utero life. During synaptogenesis, the hallmark of that developmental phase is the massive formation of neuronal synapses. Trillions of synaptic contacts are being formed, and dendritic branching occurs on minute-to-minute basis. It's incredibly fast phenomenon. In order for neurons to form proper synapses, they need to migrate to their final destination. In the process of connecting, they mature and differentiate and form this incredible network of synapses and circuitries that we rely on for the rest of our lives. What I'm talking about here is not really a formation of a few synaptic contacts when we study a different language or we learn to play piano, for example. Here I'm talking about fundamental formation of synaptic connections that we rely on for a long, long time during our lives. And of course, brain growth also happens because of the glial cells proliferation, in particular astrocytes and oligodendrocytes. And as you know, oligodendrocytes are very important for timely myelination. In humans, synaptogenesis starts roughly in the last trimester of in utero life, and then it goes on for the first 2 to 4 years of postnatal life. So it is actually a process that takes a while, but once it's over, all we have is a fine tuning and not really massive synapse formation. So what happens to neurons that do not make it to their final destination? They do not properly connect. They do not mature. They are actually considered by the nature to be redundant. And you've seen a terminology used differently, for example, physiological cell death or apoptosis or neuroapoptosis or programmed cell death. What I like the best is the term that's used by the old pathologists, and they refer to this phenomenon as a suicide of redundant neurons. These neurons are considered redundant and therefore destined to be removed. Despite the fact that this process is naturally occurring, I have to emphasize that this is not the time for the massive deletion of neurons. That happens during neurogenesis, and by the time of synaptogenesis, it's a fine refinement. So in mammalian species, we say that roughly about 1 to 2% of neuronal population in any given brain region is actually destined to die by neuroapoptosis. So that's why it is considered to be a programmed cell death or physiological, because it's tightly controlled. What is it that controls neuroapoptosis? Well, we know that two major neurotransmitters play an important role: major excitatory neurotransmitter glutamate and major inhibitory transmitter GABA. They control all key elements of synaptogenesis. So why is that important? Who really cares? Well, when you think about sedatives and general anesthetics that we use in daily practice, some of them more so than others, and all of them used at one point in time, you can see that they really belong to two major categories as far as the mechanism of action. Although we don't know exactly how they work, we are getting a pretty good sense as to what the mechanisms are. And you can see on the left column, I have GABA agonists listed. And on the right column, I have glutamate NMDA receptor antagonists. And all the intravenous anesthetics that we currently use, not only in anesthesia, but in emergency medicine and psychiatry, such as barbiturates and benzodiazepines, we use in daily practice. And in addition to that, it's propofol and etomidate, very commonly used intravenous anesthetics. In the inhalational anesthetics category, and I know Jill is particularly interested in that one, rightfully so, because it's so frequently used. We often use fluranes, and in particular, sevoflurane is very commonly used. Desflurane is kind of getting out of favor. And of course, older ones we've used in the past, but very similar chemical structure in pharmacokinetics and pharmacodynamics, is isoflurane and halothane. If you look at the NMDA antagonist group and you consider intravenous anesthetics, ketamine for sure are in that category, and ketamine is extensively used in pediatric anesthesia. And then in the inhalational anesthetics group, we know that nitrous oxide and xenon are NMDA antagonists, very similar to ketamine. So when you think about the assortment of anesthetics and sedatives that we use in our daily clinical practice in all aspects of pediatric medicine, you can see how these anesthetics could really affect two major neurotransmitters important for the brain development. So when you think about neuroapoptosis and what that means, and why I really like the suicide term that was coined by older pathologists, is that this is a phenomenon that starts, as you can see in A panel, with the clumping of the nuclear chromatin. These happen to be electron microscopy photos of ours, but you can find them in any pathology book. You can see that there is a clumping of nuclear chromatin that happens initially and the formation of what we call chromatin balls. At this point, that's an early phase, you can see the nuclear envelope is bilaminar and intact. In the later phase, the chromatin balls become actually the apoptotic bodies. There's an intermixing between nucleoplasm and cytoplasm that is normally a no-no. So you can see actually a cell that looks very sick and has a lot of problematic features, such as not only apoptotic bodies, but really very dense mixed nucleo and cytoplasm. But what is really important here and why this is a suicide or tightly controlled phenomenon is that this process occurs in isolation. This neuron does not drag million neurons down with it, just as you would see in the case of stroke or massive seizure or trauma. So this is what is so important about this process. It is tightly controlled. But because what anesthetics do is they push a lot of neurons into this redundant category, this is what you see. So we designed a very simple experiment a long time ago. We used 7-day-old rats. This is at the peak of their synaptogenesis. Exposed them to isoflurane alone or combined with benzodiazepine, midazolam, and nitrous oxide. It was a very typical anesthetic combination that we use in the operating room routinely. And I happen to have anterior thalamic nuclei, but we can look at any brain region, it would have exactly the same picture. And you can see on the A panel, that's a control animal. This is staining with silver, where dead or dying neurons uptake silver and kind of stain dark. You can see that there are spackles of black. These are dead or dying neurons that normally, naturally undergo neuroapoptosis, as we talked about a few minutes earlier. This is the panel for the animal exposed to isoflurane, midazolam, and nitrous oxide. You don't have to be a neuropathologist to see the difference. This is an enormous increase or upregulation in silver staining and in cell death. In this case, I was a fellow and I counted this as 70-fold increase compared to control panel. Now, this is the next day, 24 hours. What about 2 to 3 weeks later? So what we did is we let the animals live. We exposed them at 7 days and then let them live till they were 21 days of age. And this is at the tail end of brain development in rats. When we went to the light microscopy level, we couldn't see the difference. However, when we use ultrastructural analysis to look at the tissue and we studied the neuropil of these animals, you can see how control neuropil looks like. Massive number of synaptic contacts, very tight structure where neuronal ends and glial cells are kissing each other and really working in tandem. This is how the experimental animal looked like. Mind you, this is 2 weeks after single exposure to general anesthesia. You can see the neuropil is frazzled, empty looking, a lot of vacuolated, something that we cannot even recognize. And you go from very rich synaptic contacts to maybe one lonely synapse here, or maybe one lonely synapse there. If you look at the picture of the brain of the children who are addicted to gasoline, this is exactly how their brain looks like. So the question then is, what does that tell us? Does it really matter? So we did functional studies to study these rats. We let them live for 30 days or almost over 6 months. We put them in Morris water maze or radial arm maze that are actually memory paradigms and learning paradigms in rats. And what you can see that our control animals learn the task in a matter of a couple of days. And the task is really based on the fact that rats are excellent swimmers, but they hate to be in the water. So they try to look for the platform to escape as soon as possible. You can see that control animals take only a couple of days to figure out using the visual cues around the tank to find the platform and get out. If you look at our experimental or anesthesia exposed animals, they take twice as long. And if you give them more complex tasks, they never learn how to do it. So this was really an eye-opener for us. A lot of very talented folks got together and started looking at higher mammalian species, monkeys in this case. They exposed them to propofol. If you remember, that's another intravenous anesthetic. This is the picture of a propofol exposed animals in a fetal monkey brain with oligodendrocytes in white and apoptosis in cortical and nuclear nuclei in red, and these are neurons. You can see the neonatal brain looks just as devastated with propofol and isoforine side by side compared to controls. So not only that this neurotoxic phenomenon when propofol and sevoflurane and isoforine are used in clinically relevant concentration present in rats, but it occurs very much in monkeys. Now, if you look at the behavior of these monkeys exposed to anesthesia, in this case, it's a multiple exposures, three times over the first month or so. What is really amazing is these animals have a really problems with emotional and socio-emotional interactions with a very high level of anxiety type behaviors and aggressive behaviors as well. If watched a month later, they seem to have also some issues with motor abilities, as well as increase in anxiety in new social environments, and something that's called affiliative and appeasement behavior that is pathological in monkeys. So you can see that this is a pretty obvious phenomenon that happens not only in rats, we studied mice, we studied guinea pigs, but also in non-human primates. So what about humans? Let me just take you through 3 studies that I chose for the sake of time. So we have the very first study that came in 2009 as we were trying to grasp for what that means in human practice, because you have to remember, we anesthetized over 4 million children in the United States alone. So this is an incredibly high population of folks exposed to anesthesia very early on during their brain development and body development for that matter. So what they did, this is a Mayo study, came in 2009. They looked at the very large cohort of patients, over 5,000 kids. This is in Olmsted, Minnesota, and Mayo has a really robust database. So they followed kids that were exposed to anesthesia from 1976 till 1982, and they looked at the learning disabilities in these kids or cognitive abilities in these kids. And what they considered to be significant is when the standard scores were almost 2 standard deviations below what it could have been had it not been for the anesthesia exposure. What they did is they looked at different types of exposures, single to multiple exposures, and also the length of anesthesia exposure. As you can see, once the child is exposed more than a couple of times and for longer than a couple of hours, there was a significant tendency for having high propensity for learning and disabilities. Now, if you look at the graph form of how that looks like, it actually gets pretty scary. They put learning disability on the y-axis and they plotted against the age where they were following these kids, looking at the different learning paradigms used in school. And you can see that in kids exposed to no anesthesia, about 15% of them, as time goes on, show some signs of learning disabilities. If they were exposed once, you can see that there's a bit of a gap, but the gap closes as time goes on. If you're lucky enough to live in Olmsted County, when they're constantly testing you and providing you with remediations of different kind. If you look at the multiple exposures, and by that, I mean more than 2 for a couple of hours or so, and that is a drop in a bucket in our practice nowadays, you can see that as many as 30% of these kids actually suffer from some form of learning disability. What I find really interesting is that you can see that the gap actually widens. And why would that be is an interesting question. We believe it's because when a kid gets older, more of these complex executive-like functions are being asked of them, and they really struggle with that. Now, the study was severely criticized. First of all, it's a retrospective study, but also, if you remember, it looked at the time span from 1976 to 1982. Our monitoring of vital signs was not quite as robust. We did not have pulse oximetry, so there was a possibility for hypoxia to happen and not to be diagnosed. And also the anesthetics we used back then were very different than anesthetics we use nowadays. So this very same group actually published a study in 2017 when they looked at a newer patient cohort from 96 to 2000. They matched them to be younger than age of 3, and then you can see what are the outcomes that they were looking at, particularly focusing on learning disabilities, attention deficit disorders, and other achievement tests. So this is what they got. Again, it's a pretty good, nice group of patients. It's not 5,000, but it's about 450 in group of exposed and 460 in group that is unexposed. And you can see again a very same picture that kids exposed to single anesthetic have about 15% incidence of learning disability, a slight gap with a single exposure that closes later, and then a very substantial gap in kids exposed for more than a couple of hours, more than a couple of times. Interestingly enough, they looked at ADHD, and you can see not only that in a multiple exposed group, there is a higher incidence of ADHD diagnosis, but now in a single exposed group, there's a gap that's showing. So having kids exposed to anesthesia is really very serious. I'm going to finish clinical studies with one that came actually last year, a very new study, and actually came in JAMA Oncology and looked at the kids who were exposed to anesthesia multiple times when they had the diagnosis of acute lymphoblastic leukemia, and those kids need to be put to sleep for imaging studies as well as for the actual treatment. So what they noticed is actually this is a group of pediatric oncologists, and that's what is really interesting, is that because they followed those kids for a long time to see the effects of chemotherapy, they noticed that there was an increase in cognitive impairments in long-term survivals, and to be as high as 40%, and they couldn't really say that chemotherapy was the only reason for that. They saw the FDA warning that I'll share with you shortly, and therefore designed a retrospective study to see whether there was an effect on those kids that is due to multiple exposures to anesthesia. And this is what they concluded, and it's very interesting. They did multivariable regression models. And as far as neurocognitive impairments, they say that propofol use, and it's a cumulative dose per kilo, more propofol that was used for these kids to sedate them or put them to sleep, more likely is that they were significantly affected when it comes to global neurocognitive impairment. Fentanyl, that is incredibly frequently used narcotic in children and adults, as you go up in a dose, it didn't quite reach the significance, but think about it. This is pretty scary. Midaz, not as much, and morphine. What else? Fluorines, sevoflurane, isoforane, halothane, desforane. Very significant compared to if you think about neurocognitive impairment, and duration again comes as an important factor. So what were the major findings? They found they did the MRI studies in these kids, and believe it or not, they found white matter diffusivity in the corpus callosum. So there are some morphological long-lasting changes. They concluded that there is a slow processing speed, worse attention, and worse executive function in these kids. So FDA, as you know, issued the warning in 2016, where they basically say they actually wanted to put a black box warning on our anesthetics. There was a huge pushback. But what they're basically saying is, watch it, be careful. Don't that easily decide to put a child to sleep, and don't do it repeatedly if you could help it. Limit exposure as much as possible. And interestingly, they urge us to report if we think that there are any issues that could be relevant to anesthesia exposure. So very quick couple of slides, because I know the rest of the speakers are going to talk about it, and they are an incredible experts in that area. So I'll let them take the lead. But just to quickly to touch base on what we've done with epigenetic changes. What prompted us to do that is a very old study that came in 1981, and they looked at E4's haloten and E4, and E4 was induced when I started anesthesia 30 years ago, not anymore because it's very nephrotoxic. But haloten was in use for a long time and very similar to sevoflurane. And what they did is they took pregnant mice and they exposed them to clinically relevant concentrations of either haloten or E4. In early pregnancy, in mice, it's 21 days, so around 10 to 11 days, and late pregnancy, 14 to 17 days of in utero life. They monitor blood pressure and arterial gases to the best of their abilities, and then they studied learning performance in offspring at the age of 6 to 7 weeks. So first generation of offspring performed worse compared to controls, especially the ones exposed late in pregnancy, and that is actually at the beginning of synaptogenesis in rats. But what was interesting is that in second generation offspring that were born to dams exposed to haloten late in pregnancy and mated with non-exposed males, they showed that these offspring still struggled in learning tasks, which was a complex maze protocol that they used. So this is what made us do a couple of studies to look at the epigenetic phenomenon. One that I'm not going to have time to talk about deals with histone acetylation and the effects of anesthetics on histone acetylation. But Jill wanted me to look more at offspring. So I focused on this study for today. And we took 7-day-old rat pups. You may remember that's a peak of synaptogenesis, exposed them to sevoflurane for 6 hours, which is a pretty typical protocol. And then we looked at the DNA methylation in those rats exposed to sevoflurane and found actually that there is a reduced DNA methylation in these animals. We focused on one particular brain region that is incredibly sensitive to anesthesia-induced neurotoxicity. It's called subiculum. It's the extension of hippocampus and very important in learning and cognition. And then what we did then is we took those rats and we actually looked at their in their offspring that were exposed to those mothers, and we discovered that reduced DNA methylation was still present. We looked at some of the immediate early genes that are very much controlled by methylation and also play an important role in synaptic plasticity, Arc and JunB, and we saw that there was a significant upregulation of these genes. We also discovered that actually those changes were sexually dimorphic because it occurred only in males exposed to, born to exposed females. So our conclusion was that because there is an indication of decreased DNA methylation and that is intergenerational, that maybe that could explain, at least in part, long-lasting effects we see in not only learning and behavior, but also in behavior. So to conclude, what it is that we can take away from this: repeated and lengthy or cumulative exposures to commonly used anesthetics definitely has negative impact on young and developing brain. Learning and behavioral impairments are complex and lifelong. Long-lasting and often intergenerational genetic and epigenetic changes are being reported. And what I would really like to leave you with is we should take a great care to avoid the use of anesthetics and sedatives whenever possible. You know, if we look at the rise of the autism in this country and in the world, it cannot be only explained by the fact that we can diagnose it better and earlier. Another important possible avenue is to develop new line of general anesthetics and sedatives, and this is what my lab has been devoted to over the last few years. So quick acknowledgement to folks who've done an incredible work to make it possible for me to talk today, and all the funding that we get in order to keep our work alive and possible. Thank you so much for your attention.
Thank you so much, Doctor. That was so fascinating. And I appreciate your shedding some light on general anesthesia, because as I said earlier to you, that the field of epigenetics, the field of multi-generational inheritance, they don't tend to look at these very toxic, very high dose exposures. They tend to look at low dose ambient environmental exposures. And I find that a little bit puzzling, because I can't think of a single exposure, a single toxicant that's as potent and high dose and kind of generally neuroeffective as this one. So I'm very happy. Thank you for doing that. Now let's go on to questions. Do you think effects on the germline are direct, i.e., by binding to a receptor in germ cells, or indirect, by binding to receptors in the brain, which then has an effect on the germline?
Oh, that's a phenomenal question. I'm going to leave it up to the next speakers to talk about that. You know, when you talk about the effect on direct effect on the one who is exposed, it's most likely direct effect on the receptor. However, when you think about inter and transgenerational changes, I very much believe it's epigenetic in nature. I really do. The anesthetics are so lipophilic, they easily get into the nucleus, and then they do whatever they want to do there. And the influence on the histones and the DNA actually can be transferred from one offspring to another without direct toxicity on the neuron. The ones who are exposed, no doubt in my mind that this is really direct insult on the brain. There's no that. I mean, think about it. All these anesthetics that are ephorins, like sevoflurane, isoflurane, they're chemical cleaners. And I know Jill is incredibly interested in environmental toxicity. Think about this. Desforin is now slowly being removed because it gets into a carbon copy and it never, ever goes away. And we've used this anesthetic for decades. So can you imagine how many of us inhaled it on a day? I mean, these are chemical cleaners, environmental toxins. So initial insult is neurotoxicity. I think later one is really epigenetic in nature.
Yeah, I know that these are very small molecules. They are lipophilic, as you said, and there's nothing that stops them from entering the fetal tissues or all the body tissues that have any kind of vasculature, right?
Yep.
And entering the nucleus of those cells.
Easily. Yeah, these and think about exposure. You know, I showed the premature baby. All of these folks, we keep these little babies alive. 25 weeks is totally viable now. Premature baby 25. So think about how many moms and dads we are generating who are going to have those imprints because we are anesthetizing them repeatedly. It is a scary thought, actually.
So there's one more question, and we'll move on. What about C-section delivery? And I guess that could mean either C-section under general anesthesia or under, with a neuroaxial block. Would you anticipate effects on the fetus?
Yeah, so there were a couple of studies, you can probably look them up, they're retrospective, and they found no difference between general anesthesia and neuroaxial block, and in this case, it's usually the spinal or epidural that we do. One reason for that, I believe, is the fact that it's very short exposure, because we try to limit the amount of time from when mother is put to sleep till the baby comes out, and it's more from the OB standpoint than really that ever was conceived from the anesthesia standpoint. However, your question is, where very pertinent when you talk about maternal fetal medicine. In our institution, we do about 60 in utero operations for kids that have severe genetic, for example, diaphragmatic hernia, spina bifida. We operate in utero. These kids are exposed to high concentrations of sevoflurane to relax the uterus for 4 to 5 hours. So I'm curious to see how these kids are going to do. For the C-section, no worries there. It's too short of an exposure.
Quickly, before we go to Dr. Martiniuk, one more question. One thing I found out reading the literature is that, similar to what you just said, young infants are exposed to higher concentrations of the inhalational anesthetics. Can you discuss that for a second?
Yeah, they seem to be, strangely enough, fairly resistant to anesthetics. So we tend to give higher inhale concentration to them than we do to the adults, especially older adults. And it's a phenomenon that's been actually known for quite a while. And we are not exactly clear as to why that is the case. I think that it could be in part because moving in infants is more common than in adults, so we increase the dose. If we use paralysis in timely fashion, it may not necessarily be the case for the rest of the anesthetic.
Thank you so much. Very much appreciate your presentation.